Hi, my name is Dr. Radha Krishna. This topic is about Parkinson's diseases and microbiota. In this assignment, 4 questions were asked. The first question was choose a topic within molecular medicine that you are interested in, like genomics, proteomics, epigenetics, metabolomics. Explain the principles of molecular medicine. The second question is describe clinical practice present the latest molecular and cellular biology based on development. The third question is highlight the pathophysiology of disease, diagnostic methods, and development of the new and effective drugs. The fourth question is identify specific molecular targets contributing to a novel therapy or innovation, for example, gene therapy, metagenomics, antibody therapy, immunotherapy, artificial intelligence, and molecular modeling. Based on the first question is just a topic of molecular medicine, and I decided to choose a topic on metabolome and Parkinson's disease and its relationship and moving forward with this particular topic. Okay, in metabolome and Parkinson's disease, the molecular medicine and its applications of molecular biology, genetics, the biochemistry, and other principles to understand and treat human diseases at molecular level. The microbiomes represent small compounds metabolized of size smaller than 1,000 daltons. Metabolomics of metabolome involves the study of biofluids like sputum, saliva, blood tissues, sweat, breath, serum plasma, and organism. Metabolome is a downstream of transcriptome and proteome and is considered to be a complementary to genomics, transcriptomics, and proteomics. Identification of disease related to biofluids and the application is personalized medicine, precision medicine, and targeted identification. Metabolome and Parkinson's disease. The principles of molecular medicine can be applied in several ways, like metabolomic profiling, biomarker discovery, understanding the disease mechanism, personalized medicine, therapeutic target identification. By integrating metabolomic data with other omnic data such as genomics, transcriptomics, proteomics, we gain a comprehensive understanding of a disease biology and the development of more effective strategies to disease diagnosis, treatment, and prevention. The second question is describe clinical practices present in the latest molecular and cellular biology based on development. Latest clinical practice of the latest molecular and cellular biology based development in Parkinson's. This development is an advancement to a truly revolutionary state. Gene therapy, several gene therapies approach like aromatic L amino acids, T carboxylase, gene therapy, which converts levodopa to dopamine, the glial cell lining derived neurotropic factors which promote the survival of dopaminergic neurons. The stem cell therapies have been an exciting prospect in the development of iPSC or inducible pluripotent cells from the patient's cells, which later differentiate into dopaminergic neurons. Embryonic stem cells can differentiate into dopaminergic neurons, but needed to address ethical issues on that. CRISPR and gene editing allows precise genetic editing, which targets a mutation such as SNACA or Parkins or PARK2 or and leucine-rich repeat kinase 2 genes. These gene regulations uses CRISPR to explore the upregulating or downregulating genes involving the pathogenesis of Parkinson's and also alpha-synucleic target therapies. Targeted therapies or targeted strategies can be immunotherapy or receptor blocking. If you look at the diagram very carefully, we have immunotherapy and receptor blocking or receptor blockers, where you can actually see it can target the alpha-synucleic neurons, or there can be an autophagy clearance and aggregations. And you can actually look at heat shock protein, DNA, mRNA, okay, and many other, including like antibodies. Rifamycin, an FDA-approved drug, increases the autophagy levels by inhibiting the mTOR pathway, can reduce a dopaminergic cell death. alpha-synuclein is a protein that aggregates in the brain. Using immunotherapy targeting, alpha-synuclein aggregates and the small molecules inhibitor is to prevent the aggregation and to promote the clearance. Neuroprotective agents, these therapies are aimed at slowing down or stopping the progression of Parkinson's diseases. NRF2 activators are called nuclear factor retroderivative 2 activators, which is a transcription factor that induces antioxidant response and that protect the dopaminergic neurons from oxidative stress. More pronounced are the mitochondrial function enhancers, which are involved in the redox mechanism, especially in the electron transport chain and several mitochondrial targeted therapies. The exciting thing of personalized medicine is it's gaining traction with the bioinformatic inclusion and omnics. The treatment can be tailor-made to specific pathways and identify specific biomarkers to have effective individualized therapies, because no two persons can be the same based on the progression of disease. So advanced drug therapy delivery system, which are innovative drug delivery system that involves nanoparticles or intranasal delivery. Both are non-invasive and bypasses the blood-brain barrier and can be given in the therapeutic doses. The important thing is that to address the root causes, disease-modifying therapies address the underlying causes rather than the symptoms. An example is the leucine-rich repeat kinase 2, which can reduce the kinase activity and which is thought to contribute to the neural damage. In this diagram, if you watch very closely in the left side, where you can see the inflammatory disorder of the gut, and this gut has caused an excessive of infection, and that can stimulate trigger neuroinflammation, the pathogenesis of Parkinson's. The presence of leucine-rich kinase 2 or LRR2 that triggers the cytokines and the cytokines and the immune factors, which will go and cause neuroinflammation of the microglia, and that can cause the damage to the dopaminergic neurons in the brain, and that can lead to Parkinson's disease. If you look into the brain closer, if you look at the LRRK2 or leucine-rich repeat kinase 2, these can causes mutation and also can be related to the pro-inflammatory activity, phagocytosis, and then increased chemotaxis in the. As a result, there's a mutation of this LRRK2, which can lead to Parkinson's disease and progressive supranuclear palsy, and also can lead to other diseases, associated diseases like Crohn's disease, leprosy, and infections. So now we have a therapy called the LRRK2 inhibitor NSAID, which is very personalized and targeted therapies for Parkinson. The other therapy is antisense nucleotides. These molecules can bind with RNA and modify the expression of genes and reduces the harmfulness to proteins as shown in the diagram below. If you look at the diagram very carefully, you can see the DNA converted to a mRNA. And then you can see antisense oligosaccharides, which can bind with the mRNA and convert it into a protein, altered protein. But it can also be edited here, gene editing, and it prevents no protein synthesis. This is how to prevent damage from occurring. The third question is highlight the pathophysiology of the disease, diagnostic methods, and development of new and effective drugs. The pathophysiology of Parkinson's diseases is a progressive neurodegenerative disorder characterized primarily by the motor symptoms such as tremors, rigidity, bradykinase or slowness in movement, and postural instability. The pathophysiology of Parkinson's diseases involves a complex interplay of genetics, environmental, biochemical, leading to a degeneration of specific neuron population, primarily substantia nigra in the brain. One of the factors is a genetic factor that causes a mutation of the alpha-synuclein or SNCA, Parkin or PARK2, DJ1, LARC2 or leucine reach repeat kinase 2, causing protein degradation and the mitochondrial function and oxidative stress. Certain genetic variants and polymorphism risk genetic predisposition to environmental factors. In this diagram, if you look very carefully, there are three sections. And there's a genetic mutation, the PARK2 gets stimulated, and it causes a UPS dysfunction. It's an unfolding of protein receptors, and that causes a misfolding of proteins, and that can cause Lewy body damage. That leads to neurodegeneration. The second part will be the PINK2, PINK1, where it causes a mitochondrial dysfunction, which causes oxidative stress and a series of reactions causing apoptosis and capsia apoptosis and cause neurodegeneration. The third way is the oxidative stress can cause a new ATP depletion and can go in and develop cytokines like tumor necrotic factor or interleukin 1b or interleukin 6 and cytokines, and that can cause neuroinflammation and cause neural damage. One of the most important factor that causes that is in the development of Parkinson is the mitochondrial dysfunction. There's a reduced activity at complex 1 of the electron transport chain in the substantia nigra and impaired ATP production. This impairment mitophagy, where damaged mitochondria is selectively degraded, leading to mitochondrial dysfunction, cellular stress, and apoptosis. What happens when there's a loss of dopaminergic neurons is the hallmark of Parkinson's disease. So selective loss of dopaminergic neurons in the substantia nigra compacta. The loss of these neurons results in a decreased dopamine levels. Dopamine is a critical neurotransmitter involved in the regulatory movements, and the deficiency leads to a disruption of the balance between the excitatory and inhibitory pathways in the basal ganglia. The central point of discussion is the Lewy bodies in Parkinson's. So these Lewy bodies and alpha synuclein aggregation. If you look at the diagram, okay, the intracellular inclusion, also known as Lewy bodies, are the pathological hallmark of Parkinson's disease. The alpha synuclein aggregation is into an insoluble from fibrils, which play the central role in the pathogenesis of Parkinson's. The other central role is the neuroinflammatory or neuroinflammation and microglial activation. The resident immune cells release pro-inflammatory cytokines and reactive oxygen species, where it exacerbates the neuronal damage. The astrocytes also inhibit the functional changes and contribute to the neuroinflammation, impaired neural support. The oxidative stress, the impairment of mitochondria leads to oxidative stress, high metabolic rate. Auto-oxidation leads to neuronal damage and death. The new drug development or promising methods and the drugs are like ongentase or opicapone. It is a FDA-approved drug. It is a catechol or methyl transferase inhibitor, and it works by prolonging the effect of levodopa. Nurens or itradiphelin, an FDA-approved drug, is a adenosine A2A receptor antagonist. Another drug is a Ibrija or levodopa inhalation powder. It inhales in the form of levodopa, innovative approach, and the future direction of gene therapy, cell-based therapy like stem cells, iPSC, neuroprotective therapies like mitochondrial dysfunction and oxidative stress. The fourth and the last question is to identify specific molecular targets contributing to novel therapies approach and innovative, for example, gene therapy, metagenomics, antibody therapies, immunotherapies, artificial intelligence, and molecular modeling. So in this section, I've very specifically targeted the Parkinson disease, the microbiome, the quinolic acid, chironinin, and acyl-carnitine for the new innovative therapies for Parkinson's. Looking at the entire picture, we go into metabolome, transcription, epigenome, microbiome, proteome, and genomics. And this is the methodology I've decided to use in the approach to molecular medicine for the future of Parkinson's disease. Basically, it's a multi-omic data in the context of biology, chemistry, and the diseases. The aim and the objective, the specific molecular target I'm aiming for are the quinolic acid and the kynurenine metabolites in the Parkinson's disease. And I'm able to provide a specific microbiome for this disease. So it is basically the gut-brain axis microbiome, quinolic acid, kynurenine pathways, and the long-chain acyl carnitine. Just like any other drugs, my understanding and objective is to how does my methodology will be able to address early on established Parkinson diseases. The gut microbiome influences the neuroinflammation, neurotransmitter synthesis, mitochondrial function, intestinal barrier integrity, and the gut-brain axis, which contributes to the development and the progression of this disease of Parkinson's. To understand this, this is a pathway. If you focus on the lower part here, a shikimate pathway will ultimately break down to three components here: phenylalanine, tyrosine, and tryptophan. This is my focus, where shikimate will ultimately result in isoprenoid aromatic amino acids like phenylalanine, tyrosine, tryptophan, folate co-inhibitor, cofactors. Phenylalanine further converts to tyrosine, and it breaks down to three products of tyrosine, dopa, and melanin. We focus on this particular pathway. The next step is phenylalanine, and phenylalanine converts to tyrosine. Tyrosine converts to dopa, which is the main dopamine maker in the brain, and it can also convert to thyroxine and melanin. But if you look at the tryptophan pathway, where the kynurenine is involved, tryptophan converts to quinolic acid and then kynurenine. This is where our focus is, because kynurenine is neuroprotective and excessive quinolic acid is neurotoxic. My focus is the kynurenine pathway. So tryptophan converts to quinolic acid to kynurenine pathway, and quinolic acid is neurotoxic and kynurenine is neuroprotective. If we can measure this, then it's possible to develop therapies for Parkinson's. So the method we are going to do will be in the next slide. Our methodology is a human design. It's a biofluid based on feces, where around 1 to 2 grams can be stored at 80 degrees centigrade. We get a targeted validation tandem. So the biofluid fecal matter of 1 to 2 grams is stored. Okay, and then some precautionary methods has to be used. The use of high quality solvent and reagents, preparations of extraction buffer, sample homogenization, minimalization of the sample to the exposure to the sun and to the light and heat, and the use of inert material equipment to minimize the sample freeze-thaw cycles. So the optimization of the extract time and the temperature quality control documentation and record keeping is extremely important. So we use spectrometry. That means we take quinolic acid and kynurenine acid, and we look for a ratio between the kynureninic acid and kynurenine, and the ratio between quinolic acid and kynureninic acid, which can be measured by analytical platform. Proteomics has been used and be measured using the ultra-profound liquid chromatography coupled with a time of mass spectrometry. It's a powerful analytical techniques, and UPLC and TOFV or mass spectrometry 3, hydroquinine and quinolic acid ratio is measured using liquid chromatography, mass spectrometry, and we can also measure acyl carnitine produced in the intestine can be measured by the capillary electrophoresis coupled with the time mass spectrometry. So for the bacteria, specific identification involves the bioinformatics for this critical role of identification and characterization of the bacteria, because this bacteria is the one that produces this tryptophan. So the integration of the computational tools The biological data allows the analysis of large-scale genomics, transcriptomics, proteomics, metabolomic data, aiding in the identification. The species understanding their function and exploring within the different environment, including the human gut. So we're using the next generation sequencing, metagenomics, microbiome analysis. We use a human, that means the interface to infer the functional potential of microbiomes communities based on the metagenomic data which is existing. For the functional annotation and the pathway analysis, the Kyoto Encyclopedia of Genomes, for the understanding of the higher function and the utilization of the biological systems. So if you look at the human body, half the dopamine is produced in the gut or the intestines. So it communicates with the brain via the vagus nerve. There are certain bacteria in the intestines produces dopamine. Some of the bacteria is like Bacillus subtilis, Bacillus plantarum, E. coli or Escherichia coli. So meanwhile, the mitochondrial biomarkers are using the acyl carnitine, or we use a mass spectrometry to identify them. Bacillus reuteri, Bacillus plantarum, Escherichia coli produces CoQ10, which is a part of the mitochondrial function in the brain. Once we have identified the specific bacteria, as in the previous slide, and we needed to place the entry-coated capsules to prevent digestive enzyme from digesting or destroying the bacteria in the stomach. And we can form a 10 billion colony-forming units, which is how bacteria is measured, which is specifically can produce tryptophan. And it can all help to stabilize the chitinine and the quinolic acid ratio. And we can take it as a daily intake and for consumption. In short, once we have taken this particular capsule containing this bacteria, it will help to form a communication between the gut and the brain, and it can stimulate the alpha nucleic acid and to produce dopamine, and it protects against the neurodestructive or inflammation. And also it can also communicate with the brain through non-vagus pathways as shown in the diagram. So in conclusion, when we add a very targeted microbiome, Bacillus subtilis, Lactobacillus plantarum, Lactobacillus reuteri, Escherichia coli to produce CoQ10, tryptophan, phenylalanine, tyrosine, dopamine, which produces chitinine, putting together, we can now have an additional role on top of the fecal implant in a simplified version for the general public. So we can avoid a very complicated procedure because we can start very early in elderly population or whoever population at risk at a minimal cost. Okay, and then we can build the microbiota with the entry coated as shown in this slide. And this could be the future of prevention of Parkinson. With this, I conclude this assignment on Parkinson's disease and microbiota. I'm Dr. Radha Krishna. I want to thank for giving me this opportunity to present a future of management of Parkinson's diseases with a very low model or a cheaper model for the general public, where we can reach out to multiple thousands of people who are at risk. Thank you so much. Signing off, Dr. Radha Krishna.